Hi everyone, this is part 2 of our lecture on pathogenicity of microbial organisms. And this lecture will focus on the effect of toxins and the pathogenic effect of viruses, fungi, protozoans, and helminths. This is your instructor Kwame. So for toxins, there are two types of toxins that bacteria produce that are pathogenic. They are the exotoxins and the endotoxins. Exotoxins are protein in nature, and they are produced by both gram-negative and gram-positive bacteria. Endotoxins are produced only by gram-negative bacteria. For exotoxins, they are secreted by the microorganisms and released into the extracellular environment. So this extracellular in this context is the cell being a bacteria, or they may be released when the cell that the bacteria infects lyses. Okay. And then the endotoxins are produced, or the component of the bacteria that is the endotoxin is the lipid A part. And so if you look at the lipopolysaccharide, that's the lipopolysaccharide. The lipid A part is this part here. Okay. And if large amounts of them are released in terms of the endotoxins, it evokes a very high response or hypersensitive response from the immune system in terms of inflammation, and that can lead to endotoxic shock, where there is a fatal reduction in blood pressure. Genes for endotoxins are generally encoded by plasmids or prophages, and they work by affecting particular aspects of the host cells and or their metabolic pathway. So in terms of vocabulary, important terminologies that you need to understand. So there is toxin. So these are substances that will contribute to pathogenicity, mainly protein in nature. And then there is toxigenicity. So toxigenicity is the ability of a bacteria to produce a toxin, or ability of a microorganism to produce a toxin. Then there's toxemia. Toxemia is when you have blood poisoning. That is a result of toxins. So if you have blood poisoning as a result of toxins, that is toxemia. However, it can easily be confused with preeclampsia, which formerly was called toxemia, and which is when pregnant women would have high blood pressure, and it could result in swelling in the legs and the feet and the hands and all that. But in the context of this discussion, toxemia is blood poisoning resulting from the presence of toxins. Okay. Then there's toxoid. And toxoid is exotoxins. So these are exotoxins. Exotoxins that have been heat or chemical inactivated. So they are not active. And they can be used as vaccines to stimulate antitoxin production by the immune system. So example, if you look at vaccines such as the tetanus and the diphtheria vaccines, they are examples of toxoid vaccines. And then there is antitoxin. Antitoxins are antibodies that are specific for toxins. So antibodies that are specific for toxins. Antibodies for toxins. So if we summarize exotoxins in terms of their characteristics, their source will be gram-positive and gram-negative in terms of their relation of the toxin to the microorganism. As it grows, it produces them as byproducts. then they are released into the environment, or they may lyse and then be released into the environment. And in terms of their chemical components, they are protein in nature. Do they cause fever? No, they don't cause fever. Can they be neutralized by antitoxins? Yes. And in terms of the lethal dosage 50, that is the dosage that will kill 50% of the population of interest. You need very small dosages to effect those changes and have those results. They circulate to the site of activity, and they may affect the body before the immune system is able to elicit a response. Exotoxins may be named based on different characteristics. They may be named for the disease that they cause. So if, for example, look at tetanus toxin, it is associated with the tetanus disease that it causes. And then you have diphtheria toxin, also in connection with the diphtheria disease that it causes. Others are named for the bacteria that produces them. So botulinum toxin is produced by Clostridium botulinum. But others are also named by the action that they take, the special sites at which they act. So you have neurotoxins, enterotoxins, and genotoxins. Neurotoxins will work on cells of the nervous system. Enterotoxin will act on cells of the gastrointestinal tract. Genotoxins, note that it's in relation with the genes. So these will work on or cause breakages in the DNA of eukaryotic organisms and may lead to mutations and cancer. And examples, in terms of examples, it's important that you understand the dosages, so infectious dosage, and be able to interpret the relationship between that and whether a particular toxin is least potent or highly potent. So if you look at this example here, you have these three toxins in terms of the infectious dosage, the dosage that will cause infections in 50% of the population. If you look at botulinum, on the effect on the same size of mice, botulinum, you need just about 0.03 nanogram per kilogram size of the organism to cause the infection in 50% of the organism, of the population. The Shiga toxin, you need 250 nanogram per kilogram, and staphylococcal enterotoxin, you need 1350 nanogram. So if you're using more, it means it is less potent. So if the question is saying which is the less potent, if you're using more of this, it means it is less potent, it is less powerful, it is less effective. In terms of the types of exotoxins, you have exotoxin A and B. There are three different types. So the first one, exotoxin A and B, they are called A and B mainly because there are two polypeptides components of this exotoxin. And these are the components here, the A and B component. And these two components are assigned A and B. A is the active site of the endotoxin, the exotoxin. The active site is it's enzyme in component, it's protein in nature, an enzyme in its effect. And then the B component is the binding site. So they are synthesized from genes in the DNA, your messenger RNA, and this is the protein, the polypeptide. It's then released into the environment. This is the host cell. It has to attach to the receptors on the surface of the host cell, and it's going to be the B component that will do that. So the binding component, the B component will bind, and then it would then be taken into the cell by endocytosis, and specifically pinocytosis. So because these are exotoxins, these are tiny proteins, they'll dissolve in the fluid and they'll be taken together in a vesicle, part of the cell membrane of the host. Now in the vesicle, when they enter the cell, what will then happen is that the enzyme portion of it will then alter the cell function of the host and prevent protein synthesis from taking place. The B component already did its work with its attachment and binding, and so it will then be released out of the cell. So that's how the A and B exotoxin work. You have the active site, you have the binding site, and they all play their role. Another way or method or mechanism by which toxins, exotoxins may destroy host cells is by disrupting the membranes, host cell membranes. What they will do is that they will lyse the host cells by either making pores, holes in the cell membrane. Example would be Clostridium perfringens, or they may just disrupt the making of the protein channels. So they interfere with the making of protein channels. They could also disrupt the cell membrane that way. And Staphylococcus aureus uses that mechanism. Examples of the proteins that they use would be leukocidin and hemolysin. Leukocidin would destroy leukocytes, so it destroys white blood cells, PMN, polymorphonuclear leukocytes, and the macrophages are destroyed by leukocidin. And then you have hemolysin, which would also break down red blood cells. So if Streptococcus, which produces streptolysin, will then destroy red blood cells, not only red blood cells, but white blood cells as well. So this definitely will lead to anemia, right? And a weakened immune system. And then you have superantigens. Superantigens are a special type of exotoxin. Their effect is non-specific. So in terms of the effect they have on the immune system, they induce the immune system to release a high amount of cytokines. Cytokines are proteins that are released by cells when there is an infection, and what it does is that it controls the immune system of the host cell. However, if the antigen, which is eliciting the effects or inducing the response of the immune system, is a superantigen, what it does is that it will cause a more than normal, a hyper sensitive reaction of the immune system to the effect of the antigen. And so it's like killing a fly with a sledgehammer. So you have effects of fever, nausea, vomiting, diarrhea, and shock, and they could lead to death as well. So that the response of the immune system is not regulated as it should be. Representatives of exotoxins, this is not exhaustive, so these are just a few examples. You have Clostridium diphtheria, Streptococcus pyogenes, Clostridium botulinum, Clostridium tetani, Vibrio cholera, and Staphylococcus aureus. In terms of the type of exotoxin, based on the three that we discussed, you have AB toxin, being Clostridium diphtheria, botulinum, tetani, and then Vibrio cholera as well. Staph aureus will cause a disruption using the superantigen method or mechanism, and then Strep pyogenes will use a membrane disrupting method. Now, from our understanding of how prophages replicate, so if viruses infect bacteria, they may go the lytic cycle. They may use the lytic cycle, or they may go the lysogenic cycle. And for these bacteria, when they are infected by viruses, they go the lysogenic cycle, and that is where you have these effects of the exotoxins being produced. Then we have endotoxins. Endotoxins they are bacteria that will release toxins, but these toxins are parts of their cell wall. They will cause cell death, and in the process of causing the cell death, the endotoxins will be released. Also, the endotoxins may be released in response to antibiotics or antibodies that are produced by the host cells. Endotoxins will cause fever. Remember, exotoxins, they are no fever. So these will cause fever, and the fever is induced by the immune system. When the immune system produces interleukin, fever is a mechanism that the body uses to fight bacteria. If the body temperature is raised beyond a certain temperature for a certain period of time, then bacteria will not be able to survive. But then it has a counter effect on the host, that if it goes on for too long, it then destroys the host proteins as well. These could lead to shock when there is a fatal reduction in blood pressure as a result of tumor necrosis factor that are released as a result of the endotoxins. The tumor necrotic factor, which is part of the immune response, would also allow bacteria to cross the blood-brain barrier. So there's a barrier that prevents microorganisms from getting into the central nervous system. It keeps it sterile. Under certain circumstances, however, the immune system makes it permeable, this barrier permeable, so the phagocytes, white blood cells, can enter the nervous system, central nervous system, and take care of any infection that may be there. And now in the process, you would have some bacteria that would also cross with it. And so this happens when endotoxins are produced. There is an assay, there is a test that is used to detect the presence of endotoxins in drugs or on medical devices. And how that happens is that there is an assay called the Limulus amoebocytes lysates, and the limulus comes from the organism from which these cells are extracted. The organism is the Limulus polyphemus. So this is the genus name, and the species name is Polyphemus. So it produces white blood cells called amoebocytes. And these amoebocytes in the presence of endotoxins will produce proteins called lysates, and these proteins are clotting proteins. So based on the amount of exotoxin present, you would have a spectrum of the clotting protein that will be produced. If you have more toxins, you have more of this produced. You have less endotoxin, you have less of the lysate produced by the amoebocyte. And then it's read using a spectrophotometer to see how much of the lysate is being produced, and then make a determination in terms of the endotoxin presence. This diagram depicts how bacteria may produce endotoxin and release interleukins, and that will result in fever. So in terms of the summary for the endotoxin, you have gram-negative bacteria that produces the endotoxin. It's produced when cells will lyse, and it's present in the lipopolysaccharide, the lipid A component of it, of the outer membrane. And then it causes fever. It cannot be neutralized by antitoxin. The interesting thing is that these antitoxins may actually exacerbate or actually cause more harm than good in the presence of endotoxin. And you need relatively large amounts of them in order to have 50% of the population killed. Viruses also, when they enter cells and cause infections, may cause cells to produce interferons. Interferons will prevent viral replication, viral protein synthesis. However, almost all viruses have mechanisms by which they're able to evade the immune system and then interleukins, interferons that are produced. They may do so by growing inside the cells themselves. They may mimic important proteins, chemicals. And so, for instance, rabies virus have spikes, and their spikes mimics acetylcholine, which is a neurotransmitter. And so, it in the end, the effect that it has is that it prevents the transmission of neurons impulses to the various parts of the body. HIV will hide its site of attachment, and so CD4 cells will not be able to attach them. These are CD4 cells, the slender yellow cells here. We'll be able to find the receptor cells and attach itself to it and be able to destroy it. Now, viruses have effects on the host cells. The effects can be visualized. You can see them. And these effects are called cytopathic effects. Now, the cytopathic effect may result in the death of the cell, and in that case, it's called cytocidal, or it might not result in the death of the cell, and those are non-cytocidal effects. And it presents differently. So, the cytopathic effect may result in stoppage of mitosis, the release of lysosomes, which will cause the destruction of the cells, the lysing of the cells. It may also cause adjacent cells to join together as if they were a multi-nucleate cell. This is known as syncytium. So syncytium, this picture depicts an example of syncytium, where all the neighboring cells have joined together, and it's like a multinucleated, and it's just like one cell. This is very common in mumps disease, measles disease, and common cold as well. Then there are pathogenic properties of fungi as well. The waste products that are produced may cause symptoms. They may cause chronic infections based on allergic responses. Immune response may be hyper than it should be. Some of them will produce proteins like candida, and others have capsules like cryptococcus. And therefore, because they have capsules, they're able to evade phagocytosis. The fungi may also produce toxins. And one important toxin produced by fungi is the ergot toxin. It's the ergot, it's pronounced air, as in the air. So that's ergot. And the ergot toxin is produced by the fungi Claviceps purpurea. The ergot, which is the toxin, is found in the mycelium. So this is a 3D of the fungi. The ergot is found in portions of the mycelium, the vegetative part, which is very resistant to the environment and to treatment. And that portion is called a sclerotia. Sclerotia. This is a rye, a wheat plant, and this is the ergot fungi growing on this. So these are gnarly organisms that grow on grains, and they have been implicated in various pathogenic effects in terms of the symptoms that they produce in humans and animals as well. When food that is contaminated with ergot is consumed, it may cause vomiting, diarrhea, hallucinations, and may lead to gangrene and other serious diseases. Historically, evidence supports the fact that the 1692 Salem witch trial, which resulted in accusation of over 300 people being witches, and 19 people which were killed in Salem, Massachusetts, was as a result of ergot toxin contamination. When ergot it's consumed, it may lead to hallucination because there are components of it that are actually responsible for LSD. So it's a natural derivative, natural source of LSD, lysergic acid, which produces hallucinating effects on people who take it. In 1951, there was a case in France where the government was mainly in control of the grain distribution and flour distribution after the Second World War. And in one small town, Pont Saint-Esprit in France, there was an outbreak where a number of people, about 250 people, suffered effects of hallucination and vomiting and diarrhea, and people were doing weird things, thinking they could fly and stuff like that. And it's been connected to ergot contamination from the bakeries that were supplying bread to the residents. Protozoans such as Plasmodium may also cause pathogenic effects by just their presence or the waste that they produce. They may try to avoid host defenses as well. And they will do so like other microorganisms by growing phagocytes or by antigenic variation, where they change the antigens on the surface of their cells before the immune system produces the antibodies to fight it. A typical example would be Plasmodium falciparum. Then the presence of worms, the helminths, may also interfere with host function, and their metabolic waste may also cause diseases. And a good example would be Wuchereria bancrofti. Wuchereria bancrofti causes elephantiasis. This elephantiasis, where lymph vessels are blocked by the actions of the microorganism and leads to accumulation of lymph fluid and the swelling of the lower extremities. There are portals of exits for microorganisms when they do infect us. The portals of exits are usually the same portals that the organisms use to enter the host. And so there's a respiratory tract through coughing or sneezing. That's how they exit. Gastrointestinal tract, that's going to be feces and saliva. Genitourinary tract through urine, vaginal secretions, or penile secretions, through the skin, blood as well. And so in terms of blood, you're talking about needles or syringes and arthropods such as fleas and mosquitoes. So by way of overview, in terms of microbial mechanism of pathogenicity, there are portals of entry. You have the mucous membranes, you have the skin, and parenteral routes. And then the number of invading microorganisms is important, and that's where the LD50 and ID50 comes in. ID50. And then there should be adherents, so there are adhesins that are produced by these bacteria, these microorganisms, mainly bacteria that we're talking about. And then there's a mechanism to penetrate or evade the host system, so maybe capsules, cell wall components, specific enzymes, antigenic variations, invasins, and then intracellular growth. And then the damage that it causes to the host cells using siderophores by literally stealing the ion from the host cells, direct damage using toxins, either exo or endo, lysogenic conversion, or cytopathic effects. And then there's the portals of exit as well.